I study cancer with the goal of increasing our understanding of the inner workings of cancer cells so that one day we can design more effective drugs to treat the 1 in 3 people in this room that will be diagnosed with cancer during their lifetime. At the molecular level, cancer is caused and maintained by a wide variety of cellular proteins. One of the most prominent of these is MYC, MYC, which is the focus of my research. MYC binds DNA and turns on several hundred to thousands of genes that allow cancer cells to grow. And we know that MYC is actually overactive in 70% of all human cancers. Because of this, cancer researchers have tried to design drugs to stop MYC from functioning. Unfortunately, though, after several decades of work, no such drug exists. Fortunately, though, we are learning that MYC uses other proteins to function. One of those, much like Batman, Batman is just Bruce Wayne. But if you give Bruce Wayne a mask, a Batmobile, and his famous utility belt, he is transformed into a super crime-fighting hero. So we believe in our research that we've discovered MYC's utility belt, a protein that enhances its function. And this protein's name is MTBP. So our research indicates that MTBP binds MYC at DNA and enhances its ability to turn on genes that are necessary for growth and proliferation. In humans, we see that MTBP levels are significantly higher in cancer cells compared to normal tissue. Specifically, when we look at breast cancer, we see that patients whose cancers have very high levels of MTBP, these patients die much faster than other patients. And we know that these patients' cancers have very high levels of MYC activity. And so we know from other research that if we reduce MYC activity within cancer cells, it can cause these cells to permanently die or stop growing. And so we asked, well, if we know that MTBP is increasing MYC activity, what would happen if we removed MTBP from these breast cancer cells? And when we did this using experimental tools, we saw that by removing MTBP from breast cancer cells, they grew significantly slower and ultimately died. For this reason, we believe that MTBP is a critical partner of MYC in maintaining cancer cell growth and proliferation. Future studies that will be inspired by our work could potentially result in a drug to disrupt this interaction between MTBP and MYC, allowing to help patients that are battling against cancer. Thank you very much.